I can't think of anybody more appropriate to receive this honor to give the Don Brooks and Dana Devine address today. It's really a pleasure to welcome Karina Yazdanbakhsh again, she gives me a thumbs up on that, again to the Center for Blood Research today. Karina, we have a long relationship as co-advisory board members, so I know Karina quite well. Fortunately. She's the Senior Vice President and Executive Director at the Lindsley Kimball Research Institute of the New York Blood Center. It's a mouthful. She completed her PhD in London, UK, and then came for a short postdoc to Columbia University in Rockefeller, where she ended up staying, fortunately, as she got her first position at the New York Blood Center, and she's certainly thrived there and has made major contributions in transfusion medicine and in all kinds of aspects of hematology. She's a recipient of several major, really amazing awards, including the Outstanding Investigator from the NHLBI, and was awarded the American Society of Hematology Stratton Medal for basic research just this past year. So we're really honored and pleased to have her today. So here you go, Karina. It's all yours.
Okay, thank you so much. It's truly a pleasure to be here again, second time, and absolute honor to be giving this Don Brooks and Dana Devine Transfusion Science Innovation Address today. I was fortunately, I got to meet Dana Devine as my career was starting in transfusion medicine, but I never got to meet Don Brooks. And Dana was really a trailblazer, as we heard in our field, and she's sorely missed. So let me see. Okay, I have no conflicts with different layer. So what I hope to do today is to highlight how the importance of understanding the molecular and cellular pathways that are associated with immune pathophysiology of the underlying disease of a transfusion recipient, and how this information can help inform transfusion management of these patients. So the disease I'm going to be focusing on is sickle cell disease, and I'm going to talk about a couple of projects, ongoing projects in the lab right now that I'm kind of excited about. And one of them is focusing on how the approach that we've taken, which is kind of looking at disease pathophysiology, has kind of uncovered certain pathways that we think may be responsible for heightened alloimmunization risk in sickle cell disease. And then the second project that I hope to get to, if I can in time, is to talk to you about how looking at the immune landscape in this disease has kind of, again, uncovered a role of a certain subset of monocytes that we call petroleum monocytes in their vasoprotective role in this disease, and how, again, transfusions are intersecting with both these pathways that we've identified, and how this information, again, can be used to improve transfusion management. I'll try and touch on that. Okay, so just briefly, just very simplistically, sickle cell disease, it's a single point mutation that affects the red cell. The red cell that gets produced is sticky, again, simplistically put, sticky and fragile. It's sticky, so it's kind of blocks blood flow and results in this vaso-occlusive crisis that hallmarks this disease. And the red cells are fragile. They're deformed. They are easily destroyed. And we know that the red cells are removed either extravascularly by the reticular endothelial cells in the spleen and the liver, as well as intravascularly. They're fragile. The red cells fall apart. We have intravascular hemolysis as well. And we know that there is intravascular hemolysis because the heme scavenging removal system is overwhelmed in this disease. And the excessive heme we know that it can activate the underlying endothelium, and you have increased expression of these adhesion molecules on the endothelial cells and apoptotic markers. Think of it as a Velcro. Now your red cells and the activated leukocytes get attached to these underlying endothelium, causing the blockage of the blood flow. And so you have the vaso-occlusion, which is, as I said, a hallmark of this disease. The organs are not receiving the oxygen that they need because of the blockage, and you have organ damage. Now, due to the chronic anemia and hypoxia, you also have elevated level of this hormone, erythropoietin, which is a compensatory mechanism to increase red cell production. A little bit about transfusions. Many of these patients will receive at least one transfusion by the age of 20. Of course, the importance of transfusion for this patient population is known. It's a recommended therapy for many of their complications: stroke, acute chest syndrome, multi-organ failure. And now with what we heard this morning, the FDA approval and for gene therapy for these patients, we use transfusion as a preoperative treatment before the patient can receive this life-saving, the gene therapy, the curative genetic curative cure for these patients. Now, the downside of transfusion, of course, is that the patient can become alloimmunized. We have higher rates in this disease, and you also have the risk of transfusion-transmitted infections, as well as iron overload, but we don't understand the mechanism. The same for the wonderful, importance of transfusion. We also don't understand the mechanism. And these are the questions that at the New York Blood Center, we have a sickle cell transfusion research program that we are kind of, again, coming from the angle of understanding what the pathophysiology is, the aspects of the hallmarks of the disease, and then kind of understanding how these hallmarks are affecting downstream cellular molecular pathways that then impacts either the various immune cells. In this case would be to kind of have the negative impact of transfusion, such as alloimmunization or bystander hemolysis that we also see in this disease as a result of transfusion, but also following transfusion with hemoparasites such as malaria and babesia that we heard earlier on. Now, we're also looking at pathways, again, downstream of erythropoietin and this hemolysis, the toxic material that is released from the red cells, on how that also has a role in the hematopoietic dysregulation. That's how we think that transfusions somehow are intersecting with these pathways to really reverse this dysregulation. And again, looking at that at the molecular cellular level, and also the fact that there is macrophage dysfunction that's been described in this disease, and how our transfusions may be, again, reversing some of these abnormalities. For today's talk, I'm going to be focusing on the transfusions and the humoral response, and there are the three really people who really have spearheaded this work, and they were all postdocs in the lab, and they're now assistant professors running their own projects and their own labs. Hui Zhang, Shan Su, and Yunfeng Liu. So, as I mentioned, alloimmunization, one of the downsides of transfusion, and we have higher rates in this patient population. So we can do prophylactic matching, but not all patients make antibodies, and as a result, it's been argued that it would be costly to do prophylactic matching. You put a strain on the potential, supply logistics you're going to have, and you should really reserve. There's a school of thought that these rare units should be reserved for patients who require these antigenic specification. So my lab and several others, I'm coming from a different angle. We're looking to identify immune response of the patient to predict in advance which patients will make antibodies, these so-called alloimmunization risk factors. And that's what one of the questions I had for one of the previous speakers. What about the, not necessarily the donor units, but the recipient response? So just to, again, so EPO is elevated in sickle cell disease. These are just looking at plasma levels of EPO in SS. This is the town sickle mice. It's the mouse model we use, where the sickle hemoglobin, the globin gene, the adult globin gene, has been knocked into these transgenic mice. And we have the controls where we have the human, just normal AA. And as you can see, there's higher levels of this EPO in these mice. And so we looked at the plasma from comparing sickle to these control mice. And we identified in red these two metabolites. So we're doing metabolomics on these in this plasma and just comparing sickle versus control plasma. We saw two metabolites that were really upregulated and increased in the sickle mice. And these are now, actually the paper just got accepted, so I can say we were focusing on specifically the 20 or so amino acids, and these are two amino acids in red. Metabolite X is asparagine and Y is glycine. They were elevated. If we treated AA mice, the control mice, with EPO, the same two metabolites also showed up to be increased, kind of giving us this idea that maybe this is something to do with an EPO-driven response. And in fact, when we went and looked at the red cells, we argued that we wanted to know where is this asparagine coming from, this metabolite X, and I'll come back to you why we focused on metabolite X, which is asparagine. And we found that, in fact, compared to if you took the if you saw these AA mice and you treated them with EPO, you saw increased levels of asparagine. And the levels were, again, compared to sickle versus the control mice, the levels of this asparagine was higher. When we treated SS mice, they already have high levels of EPO, and this metabolite we couldn't see increases. But still, this is in the red cells. What was interesting was that also when we went into our patient population, again, their red cells had higher levels of this metabolite asparagine compared to healthy donors. To prove that this was coming from the red cells, we did a transfusion experiment. We transfused control red cells to sickle mice. As you can see, the levels of this metabolite, asparagine, went down in the plasma. And this was associated with decrease in erythropoiesis, right? So by transfusing them, all of this is in the paper, we have a reduction in erythropoiesis, and the level of these, the asparagine went down. When we transfused sickle red cells into control mice now, the levels in the plasma went up. Again, kind of consistent with the idea that this was coming from the red cells, the plasma levels. So the reason we had decided to focus on asparagine is because there is asparaginase that's used in leukemia setting. And so we also wanted to, and it had been shown that it targets humoral response, T cells, B cells. So we were thinking of targeting this metabolite using asparaginase to prevent humoral response alloimmunization. So we have a mouse model of allogeneic red cells we take from a mouse model that is expressing human glycophorin A on their red cells. So we take those red cells, these are our allogeneic red cells, and transfuse them into wild-type mice. And these mice were, either sickle or control mice, and these are also treated with this asparaginase. So we're removing now the metabolite. And what we saw, so if you compare the control, the gray with blue, you can see there's a drop in the allo-antibody levels. The same goes if you treat sickle mice with the asparaginase. Again, this is now comparing the red with the purple. You see a drop in allo-antibody levels, indicating that you are suppressing, in this model, this had never been done, that asparaginase can suppress allo-antibody responses. Conversely, if we treated wild-type mice with asparagine, the levels of these alloantibodies went up, again indicating that is a driver of this antibody humoral response. Taking B cells from B cells in vitro, and we have a way of differentiating them into plasma cells. If we give them this asparagine and with increase in a dose-dependent manner, we see increase in plasma cell numbers. And what we found, if we depleted them with asparaginase, now it was really interesting, was the sickle B cells that were very sensitive to the depletion effect, so that the plasma cells no longer expanded. So it prevented plasma cell maturation in this in vitro system. So the question was, what was the mechanism? Looking at it, antigen-specific humoral response is very important. We were interested to see what was going on. And so we used a model. It's just a classic model of antibody responses. It's MPCGG. And what you can do, the mice start making antibodies against this MPCGC, which is nitrophenol, and it's a T-cell dependent immune response. And what we found, again, depletion with asparaginase suppressed this antigen-specific antibody responses, and it was more interestingly, it was really more effective in the following in the sickle mice. The depletion was very effective in the sickle mice. And so going after that, looking at antigen-specific B cells and plasma cells specifically, what we found was that it was the antigen-specific B cells that were being targeted with this asparaginase treatment. And again, here we're seeing that it was the sickle were more sensitive to this depletion compared to the wild type, to this effect of asparagine on their plasma cells. So here is the working model that we have, that we're working on, that there is increased heightened levels of EPO that drives erythropoiesis. As a result, you have increased levels of this metabolite asparagine in the circulation, and this drives a B cell activation. I don't have time to tell you, but again, it's published in the paper. We've identified the B cell pathway that leads to this activation. And as a result, this B cell activation results in more plasma cells development, expansion, and this is associated with alloimmunization. If you look at this asparagine levels in plasma from comparing alloimmunized versus non-alloimmunized patients, we see higher levels in the alloimmunized, we're kind of thinking about it as a potential biomarker. And we now have a depleting agent as a therapeutic target that we're exploring as well. Okay, just quickly, I'm going to go talk about the second project to tell you a little bit about that, and it's really understanding monocyte differentiation. So monocyte, whether it's human or mice, they get produced in the bone marrow, they come out, and usually monocyte, you think of them as precursors of tissue macrophage and dendritic cells, but a subset stays in the circulation. That's how we can screen them, that you have monocytes in the circulation, and they differentiate further. And one of the questions we were asking, what is deciding whether the monocyte becomes a macrophage versus stays in the circulation and differentiate further? And we have ways of phenotyping these monocytes in the circulation, both in human and mice. We have different markers, and we can identify them. So in humans, you can really nicely separate them into 3 subsets just based on this, their surface markers. And the subset that I want to focus on are these non-classical monocytes, which are enriched in another type of monocytes called patrolling monocytes. And these have been described in the atherosclerosis and Alzheimer models where they crawl along the vasculature, these patrolling monocytes. They really require this interaction with the underlying endothelium, and they remove damaged endothelial cells from the circulation to allow blood to flow. And we had identified several years ago that these non-classical monocytes really control anti-inflammatory profiles in sickle cell disease under hemolytic conditions. So the first thing, we wanted to characterize these monocytes better, and the question was, do they really require interaction with endothelial cells? So just mixing, getting patrolling monocytes, we sort them out and mix them with either normal red cells or sickle red cells, and in the presence or absence of this HIMVAX, these are endothelial cell, microvascular-derived endothelial cells. And what we found was that these patrolling monocytes, on their own, in media alone, you don't see any uptake of these red cells. So the red cells are fluorescently labeled. We don't see uptake whether it's sickle or normal red cells. But it's only when you add the HIMVAX, these endothelial cells, and it's only the sickle red cells that you co-incubate these monocytes, sickle red cells, and the endothelium, that you now see uptake of these sickle red cells. So it was the first time to show that patrolling monocytes can scavenge endothelial-attached sickle red cells. So what they're doing, they're trying to clear up these sickle red cells that are attached, that are blocking blood flow. And what we found with their numbers were actually their frequency was lower. There was a trend toward lower numbers of them in sickle patient at steady state, but during acute crisis, their numbers really plummeted. It went down. And so that we thought that their numbers really correlate negatively with pain crisis in sickle cell. And then manipulating their numbers either by removing them, depleting these patrolling monocytes, what we found that there was increased organ infarcts in these mouse model, in our Towns mouse model, whereas increasing their numbers, you saw improvement in organ infarcts as well as stasis. This is all published now. Then our question was this question that I asked at the beginning of this section, which was like, what is deciding whether these what is controlling their patrolling monocyte numbers? As I said, they differentiate from classical monocytes, but we also know these classical monocytes also go to the tissue. And what we found, again, I just have to summarize because of time, that hemolysis plays a really important role here. What hemolysis does is that on one hand, it can drive type 1 interferon levels activation in monocyte macrophages. So we see increased levels of type 1 interferon in sickle patients. And what downstream of this interferon signaling is CCL2. This is the MCP1. This is the monocyte chemoattractant protein that is important for telling monocytes to go to the site of injury, to the tissues. And so heme does that, hemolysis does that. It drives the monocytes to go to the tissue. But heme also, we found that, also increases these are the macrophage colony stimulating factor, CSF1. And what this factor does, it drives patrolling monocyte differentiation. And it's really the balance between these two, CSF1 and CCL2, that is controlling these patrolling monocyte numbers. So we're using not only the patrolling monocyte numbers as a potential diagnostic marker for recurrent pain crisis in these patients, but we're also using this ratio now as maybe as a diagnostic biomarker for drug efficacy against pain. Now, what about transfusions and patrolling monocytes? So using, again, the sickle mouse model, transfusing them, what we found was actually patrolling monocyte numbers improved. But here, the reason it improved was that their death, they didn't die as much after transfusions. What's happening is that we found that, and again, I don't have time, this is published, that these patrolling monocytes, when they are picking up these red cells, because of the iron content, whatever, they are dying. And what transfusions does, actually, it kind of dilutes out these sickle red cells, so they're not working over time and dying. And that's why their annexin 5 percentages goes down after transfusion in sickle mice. And in humans as well, we saw some trend towards increase in patrolling monocyte frequency. So again, what's happening, we think that what transfusions is doing is really improving the survival of these patrolling monocytes. Because of time, I just quickly, again, I'm really excited about this project. So, what we think is happening, heme damages endothelial cells, these patrolling monocytes, and as a result, you have more sickle cells that are attaching to the underlying endothelium, and patrolling monocytes come up, they pick up these, this is their job, they're picking up garbage, including sickle red cells that are attached to clear the blood flow. As a result, you have this upregulation of HO1, this is a cryoprotective enzyme, and if the numbers of these patrolling monocytes are not there, they're not clearing up, this results in inflammation and vaso-occlusion. And transfusions, by improving the survival of these patrolling monocytes, is really provides a protection. And that's why we think that during many patients who have recurrent pain, they get put on transfusions. And now we have a potential mechanism why it helps. And maybe we can stratify patients who should get transfusions more or less, depending on patrolling monocyte numbers and the patient's underlying disease. So I'll stop here, and I want to thank the people who've done the work. My Yunfeng Liu is as I said, a postdoc in the lab that has his own lab. We have the research, transfusion research group, the sickle cell transfusion research group, Shu Lian, Yunfeng Liu, Cheryl Lobo, Shan Su, and Hui Zong. And we're really grateful to our clinical collaborators. And of course, none of this work could be done without funding support. So terribly grateful to NIH and NHLBI. Thank you.
Thank you. What's the receptor for the free heme that's generated upon hemolysis?
So on endothelial cells, other groups have shown is TLR4, actually. We just found that in macrophages and in the kidney, it is, and it's the macrophages, is through a TLR3 pathway that activates the downstream, and it goes in and it activates type 1 interferon signaling there. But TLR3, TLR4, yeah.
Okay. So that was type 1 interferon, it wasn't lambda interferon?
It's type 1 interferon that we found.
And so the CCL2, that was being produced by the endothelial cells?
So the CCL2, it's produced by monocyte macrophages, and in the liver we've shown that it's produced mainly by that.
But
CCL2, yeah?
But that doesn't make sense. The monocytes are responding to CCL2.
The monocyte responding to, and the macrophages are responding to the heme. Yep. They're producing type 1 interferon, and that downstream of that is CCL2.
Right, but normally for a chemostatic gradient, other cells are making the MCPs. Yes. There's 4 of them that then chemotract and activate the macrophages. Yeah. So you're talking about an in-cis feedback loop, are you?
I'm going to say that we were not able to identify the actual cell source, okay? But we think it was the macrophage. It's the same cells that are producing. That's what we were thinking. Because it's downstream of the interferon receptor 1 signaling, the CCL2 is. Yeah.
Karina, thank you very much. I'm curious about the state of reticulocytes. Yes. Are they elevated during sickle? Are they the ones that are driving metabolomics and proteomics differences during, or what you're seeing here?
So we found that the asparagine, at least in this asparagine, it is produced by these mitochondria-containing cells. So it's reticulocytes, and also it is this interesting phenomena in sickle patients. They have mature red cells that still retain their mitochondria. So I think it's the mixture of both of them. It's both the reticulocytes as well as the mitochondria-positive mature red cells.
And is that what's driving alloimmunization in disease?
So
it's extent of reticulocytosis.
Yes, yeah. So there are papers that are saying that reticulocytes do drive alloimmunization, okay? And so, yes. So the short answer, I'm just going to say yes. There's more going on because the hemolysis is also, there's another pathway going on, but that's what we think that the retics and at least in sickle cell, is driving alloimmunization. It's really interesting when you do exchange transfusions, right, where you're removing the sickle red cells and replacing them with control cells, you have lower alloimmunization as well. So it kind of fits in with that too, yeah.
So Karina, that was great. So is this a conserved, the asparagine we're talking about, is this conserved across species? And also it wouldn't be then specific to sickle cell?
Okay, so we looked at thalassemia, okay, so we did not see this increase. So I think there's something very, we don't quite understand. I think that's why it didn't get into blood. It was blood advances that got published.
Okay. And so there's no endogenous asparaginases. So are we going to sort of start squirting our patients with L-asparaginase?
So that's something where, that's what in mouse, it works in mouse. So yeah. You can prevent alloimmunization there. So you have to encapsulate it, unfortunately, this asparagine, yeah.